The peripheral nervous system consists of all the parts of the nervous system that are found outside the CNS, and of course, the PNS is responsible for both the transmission of signals from receptors back to the CNS and from the CNS to the various effectors that are found in the body. The peripheral nervous system works through these 31 pairs of spinal nerves and 12 pairs of cranial nerves that we talked about previously that emerge both from the spinal cord and the brain. And furthermore, the peripheral nervous system can be broadly divided into efferent and afferent divisions. Afferent divisions are, of course, carrying information in to the CNS, and information is carried in from all the various receptors, that we'll talk about in Chapter 7, and that information is processed, and then information goes back out via the efferent division. So if we just consider the efferent division of the peripheral nervous system, what we see is this. We divide the efferent division of the peripheral nervous system into the two divisions that are shown on this particular slide, the somatic division and the autonomic division. The somatic division simply innervates skeletal muscles, and we have a single neuron, the cell body of which is located in the CNS, and it heads out and makes direct contact, as we see on this particular slide, with a skeletal muscle, causing the skeletal muscle to contract. The contracted state of skeletal muscles depends entirely on the activation of this single type of neuron. There are no inhibitory neurons that make their way to skeletal muscles as part of the somatic nervous system, but rather the contracted or relaxed state of a muscle depends on the frequency of action potentials that head down these somatic neurons. Secondly, the second division of the efferent division of the peripheral nervous system is the autonomic division. So the autonomic division heads out and innervates things like smooth muscle, cardiac muscle, glands, and the GI tract, among a few other things in the body. And interestingly, there are a number of differences, including some anatomical ones that we'll point out here, between the somatic and autonomic divisions of the nervous system. Anatomically, what we notice is this, that in the somatic nervous system, we have a single neuron, as we previously mentioned, the cell body of which is in the CNS and the end of which is on the effector organ, like skeletal muscle. Well, it's always skeletal muscle in the case of the somatic nervous system. And in contrast, in the autonomic nervous system, once again, we have one neuron that has its cell body located in the CNS, but that neuron leaves the CNS, and then we have a ganglion. A ganglion is a cluster of cell bodies that are found outside the central nervous system. If a cluster of cell bodies is found inside the central nervous system, it's referred to as a nucleus, nuclei plural. And what we see is that we have a junction between a preganglionic and postganglionic neuron located in this autonomic ganglion. Then the postganglionic fiber will head to the effector, one of the effectors that we mentioned previously, smooth muscle, cardiac muscle, glands, and so forth, and have its impact on the body. So, to summarize, the somatic nervous system, in other words, the somatic division of the peripheral nerve, there's a lot of divisions here, aren't there? So first we have CNS, PNS, then within PNS we have afferent and efferent, and then here we're just considering the efferent, and we have somatic and autonomic. So in the case of the somatic division of the efferent portion of the PNS, we have one neuron that is used to reach the effector. In the case of the autonomic division of the efferent division of the PNS, we have two neurons that are used to reach the particular effector. Here, we only consider the autonomic nervous system. And when we do, we notice that we typically divide the autonomic nervous system into two distinct divisions, the sympathetic and the parasympathetic divisions. There are differences in anatomy, synapse location, and function. So let's look at each of these. Anatomically, what we see is this, that if we examine the parasympathetic division of the nervous system, we see that the nerves that are part of the parasympathetic division of the nervous system tend to originate either from the brain, as we see here if we look at the picture that's found on this slide, or the sacral region of the spinal cord, as we see here. As a result, the parasympathetic division of the nervous system, depicted of course in red on this particular picture, is oftentimes referred to as craniosacral because it emerges from both the brain and the sacral region of the spinal cord. In contrast, anatomically, if we look at the sympathetic division of the spinal cord, we see that it primarily arises from the thoracic region of the spinal cord and from the first two lumbar vertebrae. And as a result, the sympathetic division of the autonomic nervous system is oftentimes referred to as thoracolumbar as a result of where those neurons originate. So, obviously the sympathetic division is shown in blue on this particular slide. Secondly, synapse location. There's a difference in synapse location between the sympathetic and parasympathetic. Let's look at the parasympathetic first. In the case of the parasympathetic division of the nervous system, what we see is this, that there are long nerves, long preganglionic nerves in the case of the parasympathetic division of the nervous system, and then there is a ganglion, and then there's a very short postganglionic nerve. Take a look at that. So you'll notice in each case here that the postganglionic nerve in the parasympathetic nervous system is very short compared to the preganglionic neuron. And this is always characteristic of the parasympathetic division. Let's compare that to the sympathetic division. In the case of the sympathetic division, what we see is this, that the preganglionic neurons are typically quite short, relatively speaking. And so oftentimes the ganglions are found very close to the spinal cord itself in these rows of ganglions that we refer to as sympathetic trunk ganglia. Even if they're not, in, for instance, the case of the celiac ganglion or the superior mesenteric ganglion, then relatively speaking, and I mean relative compared to the parasympathetic, the preganglionic neuron is still relatively short and postganglionic neuron is still relatively long. Now, I think this is obvious, but perhaps I should make sure that I go ahead and state the postganglionic neuron in each case is shown by a dashed line. The preganglionic neuron is shown by a solid line. And so these dashed blue lines represent the postganglionic sympathetic neurons, and they are relatively long compared to the dashed lines we see on the other side, which represent, of course, the postganglionic neurons of the parasympathetic nervous system. So this is another difference between these two systems. And then finally, and this is most important probably, there's a very distinct difference in function between the sympathetic and parasympathetic divisions of our nervous system. The sympathetic division, as you likely know, is preparing us for a fight or flight response. And so this is an adaptive feature that we share with other vertebrates, all other vertebrates to the best of my knowledge, and it prepares us to survive a very stressful situation. You have probably personally, well, unquestionably, you have experienced the impact of the sympathetic nervous system at various points during the course of your life. I was having a discussion about this with a student recently. One aspect of your life in which you've probably noticed this is public speaking. And so it's very common for people to be nervous before they get up and give a talk in front of a group. And so you may have noticed an increase in your heart rate, an increase in your breathing rate, a bit of dryness in your mouth perhaps before you get up to give a talk in front of a group of people. Now, this is not an adaptive feature typically before you give that talk, but this is what you're observing is the activation of your sympathetic nervous system. This is a situation that makes you nervous, and your body is preparing you for this fight or flight response. And so it's a good illustration of how the sympathetic nervous system works. Now, the parasympathetic nervous system promotes what we typically refer to as a rest and digest response. And so it allows us to relax, to slow down, to fall asleep at night, and to digest food. And so if we go back to the sympathetic nervous system is diverting blood away from our digestive system. And so it's making this calculated assumption that if we don't survive the next few minutes, we're not going to need to eat anymore. And so it would rather take those resources away from our digestive system, send it to our skeletal muscles, and allow us to get out of the situation in which we find ourselves. And so that's the general way in which we should view the sympathetic and parasympathetic nervous systems, both anatomically and physiologically. Excitingly, what we see is that there are some difference in neurotransmitters that are used at these different areas that we've been discussing in our nervous system. Differences in receptors as well, I should note. So what we see is this, that let's go through these diagrams on our slide, which will illustrate this very nicely. Here we begin with the somatic nervous system, and this is something that we're all aware of, but we review once again and see that we have a single neuron that goes from the central nervous system all the way out to the effector, which is a skeletal muscle. We see, as we well know, that we have acetylcholine that's released as the neurotransmitter between this motor neuron and skeletal muscle. And we have nicotinic acetylcholine receptors that we've discussed previously in this chapter that are located on the skeletal muscle. Nicotinic receptors, as you recall, are ion channels that are nonspecific, allowing sodium and potassium to pass through and leading to depolarization of the skeletal muscle cell and action potential and then contraction of the muscle via a mechanism that we'll talk about soon. So that's the somatic nervous system and the receptors and neurotransmitter that's involved there. Now we go to the peripheral nervous system, and we see some exciting differences between the parasympathetic and sympathetic division. Let's follow the order on our screen and go ahead and do the parasympathetic division first. So in the case of the parasympathetic division, we head out to our ganglion, and at the ganglion we release acetylcholine. In fact, to make this easy perhaps, let's look all the way, go ahead and look down at the sympathetic division of the nervous system, and we see that in each case, we're always using acetylcholine at this junction between the preganglionic and the postganglionic neuron, always using acetylcholine, and we're always using nicotinic receptors. Look, nicotinic receptors, nicotinic receptors, nicotinic receptors. And so that'll be easy for us to remember. And this makes perfect sense because what we need to do is get an action potential from the central nervous system all the way down to the effector. And the only way, or the best way, let's say, to do that is going to be to use a neurotransmitter that binds to an excitatory receptor, like a nicotinic receptor, and regenerates that action potential on the second neuron, the postganglionic neuron, on this path. So, in the junction between the pre- and postganglionic neurons, we always use acetylcholine and always use nicotinic receptors. But here's where things get a little bit different. In the case of the parasympathetic division, and our postganglionic axon, we do release acetylcholine once again once we get to the axon terminals of the postganglionic neuron in the parasympathetic division. So here's acetylcholine, but look, here's something exciting. Here we have muscarinic receptors that are found on the effector, whatever the effector may be. Now this also makes sense. So keep in mind, nicotinic can do nothing but excite. Muscarinic oftentimes inhibits. Muscarinic receptors, as you recall, are a metabotrophic receptor, utilizing second messengers and having a wide variety of impacts depending on the type of second messengers that are used and what's past that second messenger. So it makes sense that we would use muscarinic receptors in the parasympathetic division to promote this rest and digest response. Now, let's go to the sympathetic division. In the sympathetic division, excitingly, at the postganglionic axon, we are releasing norepinephrine. And so norepinephrine is a totally different neurotransmitter, of course, binding to an adrenergic receptor. And as you recall from earlier in this chapter, there are a number of different types of adrenergic receptors, bringing about different responses depending on the particular type of adrenergic receptor that we have in that particular location. Now, additionally, what we see is this, that of course, the sympathetic division is promoting this fight or flight response. That fight or flight response is greatly magnified by the release of epinephrine from the adrenal medulla. And so here we see the adrenal medulla, and so I guess we should back up a little bit. So here we see a preganglionic neuron that's coming from the spinal cord and making its way to the adrenal medulla. It's releasing acetylcholine, which goes to the adrenal medulla, and the adrenal medulla is then releasing epinephrine that we see right here into the bloodstream. And so the adrenal medulla is a little bit strange. You may say, well, why aren't we talking about it here in the context of the nervous system? Well, it's actually, this is a hormone. By definition, the epinephrine is released into the bloodstream. And so, I mean, you should note here what's happening. So here, essentially, we consider the adrenal medulla a modified postganglionic axon. or neuron, excuse me, collection of neurons. Because look, here we have the exact same thing happening. We have a postganglionic neuron at the sympathetic division of the nervous system being activated, going directly to the effector and releasing norepinephrine, which binds to adrenergic receptors. Here the same thing occurs, but the adrenomedulla is activated, and the adrenomedulla simply doesn't have those axons that reach out through the body, but instead of reaching out with axons, it simply releases its product, epinephrine, into the blood. In fact, we should point out it is releasing about 80% epinephrine and 20% norepinephrine, and that goes into the blood and then makes contact with the same adrenergic receptors. Most of these adrenergic receptors are binding to both epinephrine and norepinephrine. This is going to be important for the physiology of the sympathetic nervous system that we'll talk about on the next slide as well. We close this discussion of the autonomic nervous system by reviewing and adding a few more details to the physiology of how these two systems work. Now, we've discussed the fact that the sympathetic division is preparing us for a fight or flight response, the parasympathetic division for a rest and digest response. But we want to add to that just a little bit in this particular case. So what we see is that most organs receive input from both the sympathetic and the parasympathetic division. And the overall state of the organ depends on which system is dominating at any given time. So, for instance, the heart, digestive system, respiratory rate, all these things are controlled by, you might consider them competing, by at least contrasting innervation from the sympathetic and parasympathetic divisions of the nervous system. I do want to point this out as well, though. So one last thing to point out here is that the sympathetic tends to be activated all at once. Typically, if one part of the sympathetic division is activated, then all parts are activated, with rare exception. The parasympathetic can be activated in more of a piecemeal fashion. The heart can be activated, heart rate can be sped up, but the breathing rate cannot be sped up, for instance. And that's possible physiologically as a result of those adrenergic receptors, as you recall from our earlier discussions. But it's also tied to this. It's tied to the idea that epinephrine is released in the bloodstream. And so when epinephrine is released into the bloodstream and circulates through the body, all organs are exposed to that epinephrine. So that makes it much more difficult to activate the sympathetic nervous system in the same piecemeal manner that the parasympathetic division is activated. We finished our discussion of the nervous system with a brief discussion of meninges and cerebrospinal fluid in the blood-brain barrier. The meninges are a series of membranous coverings that cover and protect the brain. So if we look at the diagram on our screen, what we see is that going in from the outside, we have the skull and then the dura mater. Just inside that, we have blood, venous blood located just below that. And then we have the arachnoid mater that's shown here. So the dura mater is the thickest of these coverings. The arachnoid mater somewhat smaller. Underneath the arachnoid mater, we have this subarachnoid space of the brain. So named because there are a number of fibrous branchings that protrude into this space that are somewhat reminiscent of a spider. And then underneath this area that's filled with cerebrospinal fluid, so this area provides something of a cushion for the brain, the subarachnoid space being filled with the cerebrospinal fluid. And underneath this, we have the pia mater, which is shown right here. The pia mater is a very thin covering that's connected to the surface of the brain itself. And so these three layers provide some protection for the brain, particularly this subarachnoid space. In the subarachnoid space circulates this cerebrospinal fluid. The cerebrospinal fluid is formed from ependymal cells, one of the types of glial cells that we discussed back in an earlier portion of our chapter. And it's formed at this region known as the choroid plexus that we see pictured here on our diagram. And so, as you would imagine, cerebrospinal fluid is formed as a result of filtering action. The pressure of the plasma, the pressure of the plasma forces a portion of it through these ependymal cells, and what's created is cerebrospinal fluid, similar to the manner in which interstitial fluid is formed in other parts of the body. Cerebrospinal fluid circulates through the subarachnoid space and other areas of the brain and spinal cord, eventually making its way to the top of the brain, where it exits via these one-way valves, making its way back into venous blood. The brain is a very demanding organ in terms of blood supply. So the brain makes up only about 2% of body weight, but receives 12 to 15% of the blood flow. The brain has no ability to store energy at all, but must be fed. The cells of the brain have no ability to store glucose, but must receive this constant supply of glucose molecules, or they start to die very quickly. And so the brain is very sensitive to a decrease in blood flow. Now, despite that, the brain is also very sensitive, and so the body is very particular in regarding what it will and will not allow to make its way to the brain. So what we're saying is that the brain needs a tremendous amount of blood, but the brain is very picky, or at least the body is very picky, regarding what it allows to exit that blood that is sent to the brain. And so we assign the name blood-brain barrier to the structures that are associated with the capillaries that are found in the brain that largely limit what can exit these capillaries in the brain. As we look at the capillaries in different parts of the body, we see that some capillaries are extremely leaky, allowing almost anything to move in and out of those capillaries. But the capillaries in the brain are the stingiest capillaries that we find in the body, that being very particular regarding what can exit those particular capillaries as a result of the need to protect the brain, and hence we use the word blood-brain barrier to describe these structures. Now, we do notice this, that being lipid-soluble, as you might imagine, means that you can get into the brain much more easily because you can pass across those plasma membranes with much less trouble. Now, we see that illustrated in drugs such as heroin and morphine. And so morphine and heroin, shown on the screen here, are very similar to one another in terms of their chemical structure, but heroin is more lipid-soluble. And so it makes its way across that blood-brain barrier much more rapidly and therefore has a much more rapid impact on physiology than does morphine. But we do see that under ideal conditions, the blood-brain barrier prevents the entry of most toxins. Metals, such as lead, for instance, are nearly entirely excluded from the brain as a result of this blood-brain barrier.